So I've realized through the communication in our previous classes that a lot of you have a doubt how meiosis can produce haploid cell during meiosis 1. So let us look at this particular question, meiosis 1 and ploidy. So this is our topic for this session. So first of all, let us look at what meiosis is in general. Meiosis is also called reductional division. It takes place usually in diploid cells, which undergo a division in two stages, meiosis 1 and meiosis 2, to produce four haploid cells, usually equally sized, four haploid cells, where with half the amount of DNA as present in the original parent. Right, so half each, half each, half each, right? So this is what meiosis is. It takes place in two stages, and both the stages are then further subdivided into prophase, metaphase, anaphase, and telophase. So let us look at meiosis 1 first of all. In meiosis 1, the cell that has to undergo division, now let us say this cell has to undergo division. First of all, let us even go before that. Now before division, let us look at this particular diagram. Before division, this is how a cell of the body is. This is how the cells of our body is. Now all the cells of our body, especially I'm not talking about the cells that are about to undergo meiotic divisions or mitotic divisions. I'm talking about non-dividing cells, like the cells of your epithelium or the cells of your heart and the brain, the ones that are not undergoing division at all. In those cells we've got 23 pairs of chromosomes. Now I cannot show all the 23 pairs because then it becomes messy, so we are just going to pick one of the chromosomes. So let us say chromosome number 1. Now this chromosome number 1 will have like hundreds and thousands of genes on it. We acquire one of this chromosome number 1 from the father, and one of these chromosomes we acquire from the mother. And this pair of chromosome is called a homologous pair. Now remember, chromosome number 1, both from the father, both from the mother, will have the same genes on the same locations. Now let us say we have a gene, let us say to produce a certain enzyme here, the same gene will be present on the same location on the homologous chromosome. If the first chromosome, if chromosome number 1, I'm just assuming, I don't know the exact number, let us assume if chromosome number 1 has 200 genes, then chromosome number 1 that you inherit from the mother will also have 200 genes. The difference between homologous chromosomes is that they may have different flavors. For example, if one of the chromosome is carrying a gene for blood group A, the other chromosome will also carry the gene for the blood group, but maybe it will carry for B, or maybe it will carry for A, or maybe it won't have for any of these two, which is called the O. So you see, homologous chromosomes have the same genes but may have different flavors of the same genes. Right. So I hope you have understood what homologous pair of chromosomes are. Homologous pair of chromosome, one is inherited from the father, the other is inherited from the mother. So just before division, what is the condition? Let us describe the condition of the chromosomes or the cells. The cells primarily. The cells are diploid, which means we have a homologous pair of chromosomes, one from the father, one from the mother. And also, what is the amount of DNA? Let us say we have got one part from the mother and one part from the father. We are not talking qualitatively here. We are not talking about the type of genes. We are saying how much. So this is a quantitative description, and we say the quantity is 2C. We got two chromosomes, right? So this is 2C. Now just before division, whether it is meiosis or mitosis, you must remember this. Just before division, what happens is the amount of DNA, not the quality of the genes, the amount of DNA doubles up. So the genome, the chromosome 1 of the father will double up, will make its exactly same replica. So we have got, this is the first and this is the replica, the opposite copy, right? So the first step just before division is called the interphase, and during interphase the homologous pairs replicate. The father's chromosome will replicate, the mother's chromosomes will replicate, and so let us say we got 23 pairs. We got 23 pairs, which means we got 23 from the father, 23 from the father and 23 from the mother. Now all of them will replicate. 23 of the father will become 46 from the father, 23 from the mother will become 46 from the mother. So here this is what we are shown. We have, to simplify it, we are just using one of the chromosome. So now what do we have? This is a homologous chromosome. This is a homologous pair of chromosome, and now we have got duplicated homologous pairs, which means that these two duplicated parts, these two duplicated chromosomes are called sister chromatids. So each chromosome has a sister chromatid now. Alright, so these are sister chromatids. These are called chromatids now, not chromosomes anymore. Now, what happens after interphase? We are specifically talking about meiosis. Now, meiosis takes place in two phases, meiosis 1 and meiosis 2. Now, meiosis 1 is important. Now, let us look at its prophase. What happens during prophase 1? So, you see, let me just rub this off here. So, the genome from the father, and let me use this. So, let's say these are the sister chromatids from the father, right? And here we have from the mother. So what happens during prophase 1 is that both of these sister chromatids, these are homologous pairs, they will come close to each other, right? These are homologous pairs, they'll come close to each other and they'll exchange a couple of segments. Some of the blue segment will go there at the same spot. You must remember, the exchange happens at the same place. So if the gene for blood group is exchanged, only blood group will be exchanged. You cannot exchange the gene for eye color with the gene for blood group. That does not happen. Right, and so if this particular section also, this might get exchanged, this might get exchanged, this might get exchanged. So this is all random. There is no logic behind it, at least none that we understand by now. So this is what happens at prophase 1. We get newer combinations, but let us just put it like this. So let us assume that after interphase newer combinations are formed. Let me put a little bit of paint around on these. So let us say this gene was this gene, and let us say some gene here was exchanged, and the same ways some gene here was exchanged and some gene here was exchanged and here was exchanged. Now after prophase, the next stage is called the metaphase, right? Let's just rub all of this and see what happens after prophase. So prophase 1 is all about exchanging the genes, and the genes have been exchanged. And then we have got the metaphase, and in metaphase the chromosomes will align. So this is the cell, and we got chromosomes randomly here. So this is a chromosome here, and then there is this crossing over going on here, and then we got some chromosomes here, okay, and then we got some crossing over going on here between the homologous pair of chromosomes. But during metaphase, what will happen is that these chromosomes will align out here at the equator. This is called the equator of the cell, okay, or we call it the metaphase. This is an imaginary plane. This is called the metaphase plate. This region is also called the metaphase plate. This is an imaginary central region, just like we have got imaginary places on the planet Earth like equator, etc. Similarly, we have got an imaginary position here. And what happens here on this on this metaphase plate? Let's just redraw the cell. Let's just complete it. The chromosomes, I'm talking about homologous recombined chromosomes, they align. And how do they align? They align like this. So let's say this is the father's homologous, this is the father's sister chromatids, the mother's sister chromatids. I'm just taking one of the chromosomes, and they align like this, okay. So there was an exchange of genes. Let us also show that. Let us say two genes were exchanged here, one was exchanged here, and likewise here genes were exchanged. And what is important for you to know right here is that this is how the cells are aligned on the metaphase. Now we will have another chromosome. Let us say we have got one more pair of chromosomes here and one more pair of mother's chromosomes here, and we may have some mother's chromosomes on this side, and this is all independently done, and we may have father's chromosomes here, and you will have to imagine that recombination has taken place. Some genes have been exchanged here, some genes have been exchanged here, right? And so these are how the genes have been, how we have got these chromosomes aligned during metaphase. Now what do you see is that the chromosome of the father is on one side of this imaginary plane and the mother is on one side. So we got father's this side and mother's this side, right? Now when separation will take place, you see, let us forget about all the other chromosomes. Let us keep it as simple as we can. So we got the father's chromosome on one side and mother's chromosome on one side, even though the recombination has taken place. But now they are here on the metaphase plate, and we have got the centrosomes here, and we got the spindle fibers, which are going to pull them apart, and eventually they will be pulled. And what will be pulled? The whole pair, and eventually they are pulled like this. Alright, so we got the pair being pulled like this, and this pair is pulled this ways, right? And you got, so what do we see? We see that the recombined sister chromatids, not homologous pairs. This is very important for you to remember. The sister chromatids are pulled to the opposite poles. You get my point? And then the cell division takes place. So now let me use the same color. And now we get two cells. We get one cell here, we get one cell here. And what do we have in this cell? We've got a pair of recombined, in both the cells we have pairs of recombined sister chromatids, not homologous chromosomes. The chromosome of the father and the mother have been separated out because of the alignment of the homologous duplicated pair in such a way. They have aligned themselves on the equator in such a way that one cell receives only all the father's chromosome and the other cell receives all the mother's chromosome only, but certain genes have been exchanged which will give rise to variations. So now this is the end of meiosis 1. We had prophase, this was metaphase, anaphase shows the separation which I have not drawn, and then we got telophase, and finally we got cytokinesis. So at the end we get two cells, right? And now what is the ploidy? Are these chromosomes homologous? Are they from two different parents? The chromosome belongs to only one parent. It's only that a little bit of gene is there, and that's why we consider this chromosome or these chromosomes as homologous chromosomes. And the ploidy here thus becomes haploid, which means the chromosome belongs in the pair of chromosomes, only the chromosome from one of the parent has been received. So for example, for chromosome number 1 we only have from the father. We may have in the same cell, we may have chromosome number 10 only from the mother, but none from the father. So you see, only one of the parent set is here, and that's why this cell becomes haploid. I hope you understood, because homologous pairs have been separated out, and homologous pairs now are not together, rather they are in two different cells, and that's why the ploidy becomes haploid. But what about the content of DNA? Now remember, here the content of DNA was 2C, which means one chromosome. We are talking about quantity, and in the interphase the quantity is 4C, but the cell is still diploid. Why is the cell diploid? Because it is a homologous pair, one father's, one mother's. But how many? What is the quantity? Amount of DNA has been doubled up, and that's why we call it 4C. So at the end of first meiosis, we get haploid cells as described a while ago, but the amount of DNA has also been halved. But what is the amount halved? It's 2C. It's 2C. And that is the reason why these cells have to undergo meiosis 2. And what happens during meiosis 2? The sister chromatids separate out through the 4 phases: prophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. These two cells will independently undergo these 4 phases, and they will give rise to 2 daughter cells each. Now each cell will receive only one chromosome, which means they have only one flavor of a gene, right? These are all haploid. They stay haploid. I hope you understood what haploid is. There is no homologous pair. There is nothing from the other partner. There is nothing from the other partner. No other chromosome from the other partner. There are no homologous chromosomes. And the ploidy remains the same, but the content of DNA now, see, there were 2 large chromosomes, and now we have only one. So we call it C, 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 C. And I hope with this video I have I would succeed in making you realize why at the end of meiosis 1 we have 2 haploid cells and not 2 diploid cells with half the amount of DNA. Thank you.